The last chapter that we are covering, that is the portion of my lectures, is metabolic integration. And so we've looked at metabolism pretty finely, down to which hydride came in and which proton was abstracted. And we looked at how the molecules of energy, in terms of what we eat, are degraded, and we make energy molecules in the form of ATP. And then we talked about how we synthesize things. But what we have alluded to in certain cases, we're now going to formally talk about, is how do we keep all the metabolism balanced within the body, and talking about different tissue types, because different tissue types actually can perform some of the metabolism that we have talked about, but many of the tissue types cannot do all of the metabolism. In some tissue types, there are absences of enzymes in the entire pathway that stops that entire pathway, and it can't go down there. Okay, where they have part of those enzymes are synthesized and in the tissue type. And so some are completely absent, and so you can't do certain things. And so we're going to look at how we keep metabolic homeostasis, which is keeping everything in balance. What we're really trying to do is keep certain concentrations at a certain level. So when you go into the doctor and you have blood work, what can't you do before you go to the doctor and get blood work done? You can't eat. You have to have a fasting blood work. Why is that? Why in the world do they want to know what your fasting state is? They want to what? They want homeostasis. Yeah, they want to check your homeostasis. They're really, really just evil people who want to put the appointment at 4 p.m. and you have to fast all day, right? No. What they want to see is your metabolism working? Okay. Can you keep the concentrations in your bloodstream right through a challenge of not bringing in the energy sources? And so one of the major ones that they're looking at is blood glucose levels, and they're looking for a certain concentration, but they're looking at other concentrations also. And so we're going to look at how it's all managed. The liver is one of the most centrally important tissue types that keep the concentrations of all the metabolites at the right level. And so it's going to package out the cholesterol and fats in little particles, right? It is also going to detect how much glucose you have in your bloodstream, and either it's going to sop it up and store it, or it's going to give it out if it's needed. We will send proteins to the liver to be degraded, and we can actually make sugar from the carbons of the proteins. And so liver is going to be a key role. Also, from the intestines, the first place in which the metabolites come in through the intestines is to hit the liver before it goes into the rest of the bloodstream. And so the liver is the first tissue that really manages all of these. What's interesting about the liver is that we call it metabolically flexible, in that it can rapidly degrade and or synthesize the enzymes necessary for different pathways very, very quickly upon detecting different concentrations. And so we will upregulate pathways, downregulate pathways very, very quickly. If I look at the important tissue types that are giving us energy molecules, liver is the central one, and it will work for, in conjunction with the kidney, the brain, the heart, the adipose tissue, and the skeletal muscle. And you can see lots and lots of arrows going back and forth. Most of these we've talked about before. Okay? The liver is going to send out energy, and it sends out the energy in the form of glucose. It sends out energy in the form of ketone bodies, and it sends out energy in the form of fats. all of which can be used by different tissue types to power themselves. Now we got to understand which ones can be used. All right, what can muscle use in terms of energy for power? Of those things that it sends out, glucose, ketones, and fats. It can use glucose, yes. It can use fats. And it can use ketone bodies. All right. Depending upon what you do, fats and glucose are probably key. Ketone bodies are also used. Okay. Adipose tissue. All right. Not necessarily thought of as using lots of energy. We actually will use glucose to create the glycerol so that we can make fats. Heart. What can heart use as energy? Ketone bodies is the primary, okay, and it tends not to use much anything else. It actually has no fat stores. It doesn't have any much of the others. It's really it can use the ketone bodies, and if mobilized in the bloodstream, it can also use fats itself. Brain, what does it use? Glucose, almost exclusively. It can use ketone bodies, but only under extreme conditions, extreme starvation. Okay. Kidney is going to be something that we haven't talked about too often, but kidney you're going to see is the second tissue type that actually can produce glucose. And kidney produces glucose mainly from amino acid carbon chains. And that's a good source because in the kidney we're going to be producing urea, and it can be excreted as waste very quickly from that particular tissue type. So 2 tissue types that actually synthesize glucose is liver and kidneys. Okay. Another thing that you may not know on this slide, LPL. It is a lipase for lipid particles. And so all of those lipid particles that we have that are carrying all the fats, there are lipases that can actually break them apart and create free fatty acids in the bloodstream if necessary. And so sometimes that is used. Liver also detoxifies organic compounds using cytochrome P450. What does cytochrome P450 do? It was something we talked about before. We talked about it in context of cholesterol. It was in the synthesis of bile salts on the way to detoxify or break down cholesterol. But what does cytochrome P450 do? Yeah, it puts hydroxides on very organic molecules. And so if anything gets in, we're going to add hydroxides to it through that pathway. All right, so that's one other major factor in the liver. Okay. Adipose tissue works directly with the liver. And adipose tissue we think of as a store of energy. The liver we think of repackaging and sending out the fats. But there is something that is really interesting that happens. There is within your bloodstream, and we talk about it in terms of the lipid carriers, in terms of cholesterol, in the bloodstream, there is always a concentration of fat, okay, being carried around, and it is being transported to and from the liver from the adipose tissue. And so we keep a concentration of fat stores that are floating around. Now, there are 2 fat stores that we can get, okay? So there are 2. 1, free fatty acids. Okay, and 2, triacylglycerol. All right. The triacylglycerol are in those particles. So we're talking LDL, HDL, VLDLs, and they're floating around. The free fats, the free fatty acids, again, aren't soluble, so what they have to be carried in is sorry, I always think of it as that, but serum albumin. Serum albumin is a protein that has it's soluble, but it has pockets for hydrophobic fats to lay down in them. And so you can either have free fats floating in those proteins carried around, or you can have the triacylglycerol. Now what's interesting is that 75% of all the fats that leave the adipose tissue, that go to the liver and that are cycling through the bloodstream, actually go back to adipose tissue. But what we do is we always keep a mobilized form of fat in the bloodstream for when it needs to be used. Okay, and so having fats in the bloodstream, a little bit dangerous in terms of cardiovascular disease, but not having energy sources readily available and I have to wait from it to mobilize from the fat, getting bound to either a lipoprotein or into the BSA and then transport, you may have died before you got that energy to you. So we balance that risk with the availability of having those fats already in the circulatory system. And so the cycle is interdependent between the adipose tissue and the liver repackaging. A couple things in terms of how often the fat goes back. It's all dependent upon the availability of glycerol 3-phosphate, and that's the glycerol by which we add the free fats to them. In this, the adipose tissue is interesting in that it has a portion of gluconeogenesis, okay? But it doesn't have all of the enzymes necessary. It can take lactate and amino acids, the carbons from that, and make glycerol 3-phosphate. It cannot make sugar because it doesn't have certain enzymes. But inside the adipose tissue, we're going to use glycerol in the formations of fat. The other way that we can get the glycerol, you would think if you broke down triacylglycerol, you get glycerol. You think you can reuse it. The problem is inside the fat, we don't have the enzyme to take glycerol and put it back here. That's missing. Once glycerol is formed, it has to go through the bloodstream. It can be resynthesized back into glycerol 3-phosphate inside the liver. And we can also use either glycolysis or gluconeogenic intermediates to get glycerol 3-phosphate to make the fat back in the liver itself. And then that can be repackaged and put back. So the availability of how we make glycerol 3-phosphate is important in how we move the actual fat between the two. Muscle. Okay, muscle is where we use a lot of energy. And we can measure energy consumption by the consumption of oxygen between resting and fully active. It can be as much as 90% of the oxygen consumption. All right. Think about muscle and where energy comes from. Okay, muscle itself, resting. The primary source of energy is from fat. We will break down the fat and use that to power what's going on. Okay? Activity becomes a little bit different. Okay? If it is quick, meaning somewhere in the seconds, like 1 to 5 seconds worth of activity, what is the energy source that we're going to use for contraction? inside the muscle? Not glucose. What is your primary energy molecule? ATP. We got enough ATP to do a very quick amount of exercise. Okay. If we go a little bit further, let's say 1 minute of activity, okay, then we're going to use something else called phosphocreatine. We will learn about that in the next slide. I'll show you that. Okay. Between or after a minute to about 1 hour, we're going to use glucose stores in the form of free glucose and glycogen. Okay. And we would start to transition 1+ hour. We start to transition and we use less glucose and we start to transition into fats and then amino acids. And so depending upon what you are going to do, we're going to transition through different kinds of energy when we are trying to produce ATP. Now, on top of that, you have 2 different pathways. You have aerobic and anaerobic. Aerobic allows us to produce CO2 and the majority of ATP, but under very constrained conditions, you can form lactate and you get a little bit of energy. All right? And for muscle contraction, we need ATP. And so all of these sources will be used to produce ATP. And hopefully we're doing it aerobically. All right? If we go to lactate, we call that fermentation. Now, we got a little bit of concentration of ATP when we have muscle, okay, in the muscle itself. And so here is an NMR from a phosphorus source, and we see a peak at alpha and beta and gamma. What are we looking at here? The phosphates of the ATP, right? All right. And we look at the NMR of during exercise. Okay, NMR can be done while something is moving. And we can see the relative beta, alpha, gamma. When we break ATP in muscle, what do we produce? If you go through all the steps of muscle contraction, the myosin head, it binds ATP. What is released? ADP, right? And a phosphate. Okay? Well, take a look at this. The relative concentrations of the gamma, alpha, and phosphate to this, the relative concentrations of the gamma, the alpha, and the beta during exercise. What do you see? They're about the same. We don't see this big rise in ADP. Okay? But what do we see in terms of the change of a peak? PI goes up, right? I am breaking off 1-phosphate. There's PI. That indicated that I did do exercise. There's this peak here, phosphocreatine, that goes down really, really pretty dramatically. All right. If you know someone who works out and they go to GNC, one thing that they can buy is creatine, right? But how do they sell a creatine? Do you get a little bottle of creatine? No, they get it in the big jugs, right? And you take that big bottle and you got this scoop and you take the scoop and you put 1 and 2 and 3 and you stir it all up, okay? Why in the world are they doing that and putting that in? So you drink that. I have no idea what this tastes like, but you drink it And what are you, and it goes right to the liver, and what are they trying to do? They're trying to get creatine inside the cell, okay? And if it gets inside the muscle cells, it can be phosphorylated, okay? Why do you have to have this big old bucket? Is that the normal way you get creatine inside your cells? No. You flood your system with all of this, right? And you allow it to leach into the muscle cell, and you get a fraction, okay, of the creatine inside the muscle cell. Now, what that fraction is could be percentages, maybe less than 10%. Okay, so you buy that big bottle, that big jug, and you get a little bit in your muscle cell. Where does the rest go? You flush it right down the drain, okay? But you got more creatine in your muscles, okay? Why in the world would you do that? Yes? Vitamins? That is true. Because you take 1,000 times the recommended daily dietary allowance every day, okay? But in terms of what you're trying to do is you're trying to supplement and have enough go in. So if you can get 1% of 1,000 times, you're good to go, right? And so that's great. Okay, so I've got all of this phosphocreatine now, or the creatine and phosphocreatine in my bloodstream and in my muscle. All right, what does that help people do? What are they trying to do in this? Not that they can recover faster. They can work out longer, more intensely. Okay? And so, if you use up all your ATP, then you shift over to this. It's a real quick conversion back to ATP, and you work out longer. You bulk up. Okay? Not only do you bulk up because you work so hard, but is that polar or nonpolar? That molecule? Polar. So when it comes in, what does it bring in with it? Water. So your muscles bulk up. Okay? Some of it is water gain. Okay? And some of it is building because you can work out longer. Oh, you can. Now, in this case, this is not, well, check this out. It's natural. Because it's a molecule that's already existing in the bloodstream, right? So it's a natural product, all right? What kind of, if you have more in your muscle, you do have the ability to work out longer than if you didn't, okay? What kind of consequences, though, can you have with doing this over a long period of time? Your body, well, it becomes dependent if you want to work out at those lengths of time. One of the things that you have to watch out for is kidney disease, okay? Because it's filtering out, you're challenging your bloodstream with a lot more stuff than should be there. Yes?
Just from experience and being around people's muscles tend to tear and rip.
You can work out so much that you overstretch the limitations, and there is the homeostasis about how much you have in your cells is balanced by how much work you should be doing, and if you extend that work, you can cause damage at the same time. Although there is micro tearing actually promotes building of muscle, and so if you are trying to bulk up, that is a method, but people may overexert themselves and actually cause damage that particular way. Yes?
So is taking creatine actually effective?
Effective? You can build muscle mass, yes. You can work out longer, yes. Could it cause you to work out too long? Yes. Are you challenging your kidneys with things that you shouldn't be doing? Probably. Is it healthy for the rest of your life forever? Probably not. And as soon as you stop it, bulked up, as soon as you stop, what happens? It leaches out and it takes all the water mass away, and so you shrink up really quickly too. And so the lasting effects are dependent. Yeah, you just shrivel up. The lasting effects of these types of things are present when you're doing it, and maybe not sustainable once you stop. Okay, almost like a diet, losing weight when on the restrictive diet, and not losing weight when you're not there. Okay, and so people do use this, but it is the fastest way that you can restore ATP once it's broken. If by chance you do a anaerobic workout and you do create lactate, lactate is a dead end product inside the muscle cell. It has to be leached out, and it will leach into the bloodstream, go to the liver, and lactate is a source of carbons that can be resynthesized through glucogenic pathways into glucose. This is called the Cori cycle, and that's C-O-R-I, the Cori cycle. Okay. Here's something that is interesting. Muscle. It lacks fatty acid synthesis, the synthase. That means it can't synthesize fats. It also lacks glucose 6-phosphatase. So that means it can't export glucose. And so if any acetyl-CoA gets inside, it's going to be broken down. If any fat gets inside, it's going to be broken down. If any ketone body gets inside, it would be broken down. If any glucose gets in there, it can be stored for a little bit as glycogen, but it will be broken down. The muscle doesn't allow much of the energy sources to be sent out. The only time that there is energy molecules coming from the muscle is during muscle breakdown. The actual protein, the carbon chains of the proteins can be used to synthesize other entities, of which when you do get into starvation to allow your brain to function, you will be breaking down muscle to make sure that glucose is being synthesized. Glucose in the brain, that's the main pathway that we try to use. Under starvation conditions, the brain can shift over to the use of ketone bodies. All right. 60% of glucose utilization comes from this organ in resting patients, and it's again only under extreme conditions. What is interesting is that fat cannot pass the blood-brain barrier, so the brain cannot use fat stores. The brain cannot convert the amino acids into glucose, so it can't do that. It will allow glucose to pass, and it will allow ketone bodies to pass the blood-brain barrier. So those are the only two molecules that can be used. And the brain does use beta-hydroxybutyrate as the ketone body that it uses. All right. It will create ATP, and the reason why we have all the ATP synthesized is for the sodium-potassium ATPase. What in the world does a sodium-potassium ATPase do for us in the body? When did we learn that particular enzyme? It does. What does that enzyme do? Yeah, it's one of these things. It's if I transport one way and the other, that's what port? Antiport. And how many sodiums go out? 2. How many potassiums go in? Okay. And that creates a gradient. What kind of gradient? It's 2 charges out, 3 charges in. It's a it's an ion gradient, it's an electrochemical gradient, yes? And that's all involved in what? If you create a gradient, you can break that gradient and you allow the ions to flow back, and you get what across the membrane? What? An action potential or depolarization. And so the brain, that's all it does. It doesn't contract. That's not where it uses its energy. It is pumping ions because we're thinking. And that's all electrical impulses. And that's what we use all of that glucose, all of that ATP for, is to regenerate the electrical impulses after depolarization. Now, all of this is carried around in the bloodstream. For most adults, we have about 6 liters of blood. And mainly, blood is water that has things dissolved in it. All right. A lot of times we do tests, and this is the only time, or one of the only times that I've ever used, you memorize this, but what is this concentration right here if I wrote this? Deciliter. That's not a conversion we do very often. Okay. A deciliter is 100 milliliters. We do use that concentration or that volume in several metabolites that we measure in medical conditions. And so a lot of times you want to have a number that is below 100 when I talk about glucose concentrations. And what that really means is milligrams per 100 milliliters. If you go above that threshold, you start to talk about your body as not keeping the concentration of glucose correctly. And if you go way below that, that's also a disease state in which there's not enough energy in the bloodstream. And so we try to keep it somewhere below 100, but not lower than 70. So once you start to get past 70, you start to see physiological effects. All right, mostly water. And then we have cells. The 3 main cells, erythrocytes, leukocytes, and platelets. What do the erythrocytes, what is that? Red blood cells. And what does it do? Carries oxygen. Interestingly enough, the red blood cell doesn't do the full glycolysis, and the only way it makes ATP, it can't go through the oxidative phosphorylation. The only way it makes ATP and energy is through the glycolytic steps. What are leukocytes? Red blood cells. And the platelets are? And they are involved in clotting. All right. The other stuff that is dissolved, you've got inorganic compounds. So every salt we talk about. So if you sweat out, these are electrolytes. All those ions are electrolytes. And so if you sweat enough, then you need to have an energy drink to replenish your electrolytes, right? And so Gatorade is an example. Do you know where Gatorade was formed? University of Florida. Football team. Yep, and that's for the Gators. So if you now know that, you may not want to drink it from this point on. Okay, other organic metabolites that are there, glucose, obviously, we have amino acids, and all the things that we've talked about, lactate, pyruvate, ketone bodies, urea, are dissolved in there. And then we have a bunch of proteins, which include all of the particles that we've talked about, immunoglobulins, which are part of your immunity. There are proteins that are part of the cascade for clotting. There are other proteins that transport. And so the serum albumin is there that carries around fat. And so all of these are dissolved. It is a solution that has a lot of function. And what we're really trying to do in this metabolism is making sure the balance of those and the balance of these are in the right concentrations. All right, we are going to also talk about some control points in terms of epinephrine and norepinephrine. They're all catecholamine hormones. What do you know about epinephrine itself? Yeah, it's a signal for starvation state. It's a common name would be adrenaline, fight or flight. Okay, where do you think these molecules are derived from? What source? What structure is very similar to these? It looks like what? Hmm? No, it doesn't look, well, there's a part of it that looks a little bit like acetylcholine, but no, it isn't made from acetylcholine. No, it's not a steroid, although you do have a ring structure. Is there anything that you have a ring structure that has hydroxide on it? It looks like an amino acid. Which one? Tyrosine. These are tyrosine derivatives. Okay. There's a whole family of tyrosine derivatives that are signaling molecules. We'll take a look at some of those in just a moment. Epinephrine and norepinephrine are fight or flight signals, and they affect the muscle, adipose, and liver tissues. And what we think about fight or flight, okay, you think about this. I need energy. So any pathway that breaks things down to make ATP probably is going to be faster. Okay? I need energy. I do not need to use energy. So it's going to tend to, for most pathways in synthesis, shut them down. Okay? Also, not only do I need energy, I need that to get around the body faster. So you're going to have more flow of blood throughout the system. And so those are the main things. So if you take a look at physiologically, your heart rate goes up, your pressure goes up, and you dilate your respiratory passages. Heart rate, moving stuff around, much more. Blood pressure, well, if I'm moving stuff around, you get more pressure. Why in the world would I dilate my respiratory passages? Breathe more, bring in more oxygen so you can use energy efficiently. All right. Glycogen breakdown, muscle and liver, that seems to be good. I want to mobilize glucose. Glycogen synthesis, muscle and liver, I don't want to store. Gluconeogenesis and liver, this is backwards from what you would think, but liver will synthesize, send out glucose to the muscle and the brain. Glycolysis in muscle will go up. I want to make energy. Fatty acid mobilization in adipose tissue, another energy source. Glucagon and insulin, we're going to talk about those in just a minute, but can you give me a preview? Insulin is released when? Well fed. Glucagon is released when? Starvation. Do you know what glucagon and insulin are physically? They're a molecule in the bloodstream that is released. Do you know what they are? Is it a small molecule? Is it a big molecule? We use them all the time, but we don't think about it. They are peptide hormones. So they're small peptide chains that are hormones that are released. All right. And so insulin goes down because insulin tends to do synthesis. Glucagon goes up because it tends to do breakdown things. And so norepinephrine and epinephrine work together similarly to glucagon and are opposite to what we see with insulin effects. Okay? They are secreted in the brain and sympathetic nerve endings, and epinephrine can also be excreted in response to low blood glucose level, not only fear. The function is to make sure that glucose is in the blood and we can use it. All right. Where these are released, both of them are, glucagon and epinephrine are released by the pancreatic cells. The pancreatic cells have alpha cells and beta cells. The alpha cells will detect and release glucagon. The beta cells will detect the level of glucose in the bloodstream, and it will excrete insulin. What is interesting about the pancreas is that the pancreas is always excreting both glucagon and insulin. It just raises one or lowers one. to change your metabolic state. And so it's not on and off. It is a balance between the two. Okay, once insulin is released, it affects lots of things. Oh my goodness, that looks really, really, this looks like a wonderful slide, doesn't it? All right, what in the world is going on here? Okay, let's take a look at skeletal muscle. The main thing that you need to know is that insulin will bind a receptor. It will cause a cascade event, and we get a protein phosphatase active. So it starts to dephosphorylate lots and lots of things. Now, we've learned each one of these enzymes in the past. We now have to remember what in the world that does. Okay, what is phosphofructokinase 1 involved in? In which pathway? Glycolysis. And if I make that one positive, I make it faster, what happens to glycolysis? Speeds up. All right. It also will speed up glycogen synthase. What does glycogen synthase do? You got it. Makes glycogen. All right. What is the molecule source for glycolysis and glycogen synthesis? What starts glycolysis and what starts glycogen synthesis? Glucose. Yeah, it's like really simple, right? But glucose, I break it down for energy, and glucose is being sent to glycogen synthase to do what? Store it. I'm breaking it down and storing it at the same time. In a well-fed state, when you have lots of glucose coming in, you want to do both of those things at the same time. All right? This allows us to have a very, very high glucose level in the bloodstream, but then bring it down to a normal level very quickly. One of the reasons why we don't take blood right after you've eaten a meal is you may have a blood glucose concentration of 300 after you eat the Snickers bar. Now, if you have a glucose level of 300, most doctors would be thinking diabetic coma, right? That's only if it's at 300 for a very long extended period of time. You eat that Snickers bar, all right, and the blood glucose and the concentration goes up very quickly and then comes down very, very quickly. So because it's being utilized very, very quickly. Okay, phosphorylase kinase. Do you know what that enzyme does? It will work on, it's a kinase that works on phosphorylase. And it stops it. Glycogen phosphorylase. What is that? It actually breaks down. So this one right here regulates that one. So that gets turned off, and the glycogen phosphorylase gets turned off, which then turns off breaking down glycogen. All right, so I get all of the make glycogen, store, and I make glycolysis work. All right, what does the pyruvate dehydrogenase complex do if I upregulate that? Takes what? Pyruvate and makes what? Acetyl-CoA. And acetyl-CoA then can go where? Either the citric acid cycle or fatty acid synthesis. In this muscle, where will it go? Fatty acid synthesis, or will it go through glycolysis? Has to go to glycolysis. Why does it have to go to glycolysis? We don't have the fatty acid synthase, so it must go to glycolysis. Also, what is GLUT for? Glucotransporter version 4. That is very good. Glucose transporter version 4 is normally not in the plasma membrane. Normally it's sequestered, but when high concentrations of glucose arise, glucotransporter 4 is moved to the membrane and it will bring in and suck in a lot of glucose into the muscle cell. And so that is upregulated. All right. If I take a look at adipose tissue, GLUT4 goes to the cells. That's great. It starts bringing in lots and lots of glucose. And what we have is we also upregulate lipoprotein lipases, which then break apart the fats that are floating around in the bloodstream. And these two will promote the synthesis of the triacylglycerols, and it will downregulate the hormone-sensitive lipase, which then stops breaking down fat. And so when I'm well fed, I want to store fat. Insulin, when it gets to the liver, okay, kind of does real similar things. Phosphofructokinase does the same thing. Glycolysis is working. Glycogen synthesis is up. It's working. The phosphorylase kinase goes down. That's good. Glycogen phosphorylase goes down. That's good. I stop the synthesis of glycolysis, and I downregulate fructose 1,6-bisphosphate. Okay? And what we do with that is we tend to make glycogen synthesis and fatty acid synthesis under the well-fed state. In this, glucokinase is upregulated. Glucokinase is similar to what other enzyme that we learned? Glucokinase was compared when I introduced what enzyme? I introduced it with hexokinase. What's the difference between hexokinase and glucokinase? It is in the liver. Yes, hexokinase is in other places. What is the other difference between those two? Hexokinase will be almost always active because of its low Km. It doesn't take a lot of glucose for it to start phosphorylating. Glucokinase doesn't work very much until there's a very high concentration of glucose in the cells. And so when you are in the well-fed state, that particular enzyme is actually upregulated. The pyruvate dehydrogenase complex is also upregulated. We also upregulate protein phosphatase 2A, okay, which causes this enzyme to work. What does this enzyme do? It is the committed step of what? Of what? Fatty acid synthesis to make malonyl-CoA. And so I make fat, I make glycogen under these conditions. And so insulin will affect lots and lots of players. The last thing, there are in the brain signals that will say, hey, I'm well fed. Reduce my hunger, and I don't want to reduce my hunger, and it will stop the molecules that send out, hey, I'm hungry, start eating. All right, and so that's what insulin does. All right, we will continue on. We'll take a look at what glucagon does next, and then we'll talk about how we use energy in terms of its rate